In this lecture, we're going to step back from our present theme of development and homeostasis in order to consider how the knowledge that biologists have gained over the last several decades about how genes work has led to our current revolution, which we refer to as genetic engineering and biotechnology. It seems like barely a day goes by without us hearing some news story having to do with manipulating genes, moving genes from one organism to another, or the impact of these kinds of genetic manipulations on business, medicine, or agriculture. If our predecessors in the 20th century were living in the electronic age, I think it can truly be said that we in the 21st century are living in the genetic age. This is an appropriate point in this course to take this detour because we've now learned most of what we need to know to understand what genetic engineering and biotechnology are all about. In many ways, the key points we need to understand are really quite simple and stem mostly from the original description of the DNA double helix that was proposed by Watson and Crick, and more specifically, stem from the complementary base pairing rules that they deduced a half a century ago. Remember, with A always pairing with T and G always pairing with C. In a very real sense, advances in genetic engineering and biotechnology have had as much to do with developing new ways to apply what was learned long ago. In today's lecture, we're going to take a quick look at some of the most important techniques in biotechnology, and these are first, how to cut and paste a molecule of DNA, and second, how to move DNA from one organism into another organism. Let's first take a look at how you can cut and paste a molecule of DNA, or to use the more technical term, how you make recombinant DNA. Now, we've run into the word recombinant already in this course when we talked about the mixing of genetic material that occurs during meiosis through independent assortment of different chromosome types and through the crossing over that occurs between homologous pairs of chromosomes. In that context, we used the expressions recombinant genotype or recombinant phenotype to refer to cases in which traits from different parents were mixed in novel ways. The expression recombinant DNA generally refers to the same thing, the combination of DNA from two different sources. But there are two important differences. The first difference, of course, is that when we make recombinant DNA, we determine which different pieces of DNA are being brought together to form a new combination. Unlike genetic recombination in meiosis, where the pieces that come together, which pieces come together, is largely a matter of chance. The second major difference is that we're able now to combine DNA from completely different organisms if we choose. Our ability to create recombinant DNA in the lab is based on a fortuitous discovery having to do with how bacteria defend themselves from being attacked by viruses. Remember that many kinds of viruses specialize in attacking bacterial cells. We call these bacteriophages. These viruses attach to the outside of the cell and inject their DNA or maybe their RNA into the cell. And then the virus's genetic material essentially takes control of the cellular machinery of the bacteria, turning it into a factory for making more copies of the virus. Of course, this is very bad news for the bacteria, so you can imagine that natural selection would favor the evolution of mechanisms that defend the bacteria against viral attack. One such mechanism, discovered in the late 1960s, involves enzymes produced by the bacteria called restriction endonucleases, or we sometimes call these simply restriction enzymes. These restriction endonucleases cleave the double helix of the DNA molecule, breaking it into two pieces, and in so doing, render the DNA non-functional. In this way, the viral DNA that's injected into the bacterial cell is essentially chopped up before it can take control of the bacteria. Well, this is an effective defense, but you might see a problem here. If restriction enzymes produced by the bacteria can cut up a molecule of DNA, what's to stop these enzymes from attacking the bacteria's own DNA? Part of the answer to this question is exactly what's made restriction enzymes so useful to molecular biologists. This has to do with the fact that a restriction endonuclease doesn't cut the DNA double helix in random places, but instead only cuts the molecule at a precise point defined by a particular sequence of bases. These sites are called recognition sites or restriction sites. Restriction endonucleases are so named because they only cut the DNA at these so-called restriction sites. Restriction sites are typically only a few nucleotides long. In fact, usually only about 4 to 6 bases long, although some can be longer, maybe up to 20 bases. Now, another interesting characteristic of restriction sites is that the two complementary strands of DNA usually are palindromic with respect to each other. Now remember, in language, a palindrome refers to a set of words or letters that spell the same thing backwards or forwards. For example, the word dad is a palindrome, as is the sentence, Madam, I'm Adam. For DNA, the palindrome occurs when a palindrome occurs when the two strands of the double helix have the same sequence of bases in reverse direction with respect to each other. Thus, if the sequence of one strand is GAA TTC, then the complementary sequence on the opposite strand would be CTTAAG. It has to be this because of the complementary base pairing rules that Watson and Crick discovered so long ago. Because CTTAAG is GAATTC backwards, this sequence represents a palindromic stretch of the double helix. Now, before we go on to talk more about how restriction enzymes work and how we can use them, let's go back to the problem that the bacteria has. How does the fact that these restriction enzymes only cut DNA at a specific palindromic sequence help prevent the enzymes from chopping up the bacteria's own DNA? Actually, this fact doesn't help the bacteria directly because you can imagine, given that these sequences are so short, it's very likely that somewhere in the bacteria's own DNA that sequence will occur in one of its genes. However, what the bacteria can do is to selectively protect the restriction sites found in its own genome by slightly modifying the bases at those sites. Specifically, what happens is that so-called methyl groups, these are single carbon atoms bonded to 3 hydrogen atoms, are added to some of the bases occurring at the restriction sites of the on the bacterial's own chromosome. This so-called methylation prevents the restriction enzyme from identifying that area as a restriction site and thus protects the bacteria's own DNA. So, now that we've discovered restriction enzymes and how they work, each of which cuts a double-stranded DNA in a different place, how does this help us make recombinant DNA? Well, the answer to this question relies on one more fact we have to learn about how restriction enzymes work. Not only does a restriction enzyme recognize particular sequences, these short sequences of nucleotides, it also cuts the DNA strands very precisely between just 2 particular nucleotides in the sequence. And because the restriction site is palindromic, the exact place where the cut is made on each of the 2 complementary strands of the double helix will be offset from each other by a few bases. Well, to understand this, let's go back to the same example of a restriction site I gave you just a minute ago. If the restriction site sequence of one strand of DNA is GAATTC, then we know that the sequence on the opposite complementary strand will be CTTAAG. A restriction enzyme recognizing this site might, for example, cut the DNA exclusively between the G and A nucleotides. In this case, then, after the double strand is cut, each cut piece will now have a short section of single-stranded DNA extending beyond the double-stranded portion of the molecule. Picture in your mind these 2 sequences lined up one on top of each other, which is the way we might draw a flattened version of the DNA double helix. Now, picture cutting each sequence between the G and the A nucleotides. On the top strand, we'll have a single G on the left side of on the left fragment, at the end of the left fragment, and the sequence AATTC on the right side of the fragment, while on the right side of the cut. While on the bottom, we'll have the sequence CTTAA on the left and a single G on the right. The sequences AATT on the top strand in our drawing and TTAA on the bottom are the single-stranded ends produced by the offsetting cut made by the restriction enzyme. The exposed bases on these single-stranded pieces of the cut DNA molecule are called sticky ends. Now, the reason they're called sticky ends is because these single-stranded ends will form complementary base pairs and essentially stick to any other single-stranded sequence of bases having the complementary sequence. And here's the key point. The sticky ends from any fragments of DNA that have been cut using the same restriction enzyme will always be complementary to each other by definition. This means that if you cut two different DNA double helices with the same restriction enzyme, even DNA from completely different species, when you mix all of those fragments of DNA that occur together, they will come back together by the sticky ends base pairing with each other in different combinations. So you can mix and match DNA in this way. Then, if you add to the mixture the enzyme DNA ligase, this enzyme stitches together the newly joined DNA helices by building back the strong chemical bonds that the restriction enzyme had broken. Remember that DNA ligase is an enzyme normally that we were introduced to when we were talking about DNA replication. This is the enzyme that stitches together the pieces of DNA produced on the so-called lagging strand during replication. Once you've used DNA ligase to put the sticky ends back together permanently, you now have a piece of recombinant DNA that's made up of two different original molecules of DNA that were cut originally by the same restriction enzyme. Well, there are hundreds of different kinds of restriction enzymes that have been isolated from a variety of bacteria, and each different kind of restriction endonuclease recognizes a different DNA sequence, and thus cleaves DNA at a different point along the overall length of a molecule. By using different restriction enzymes, you can cut molecules of DNA in different places and then paste them back together in different arrangements. Now, of course, in practice, there are many, many other details that you have to consider to make this process work. But in theory, at least, our ability to cut and paste DNA to make recombinant DNA all boils down to the use of enzymes normally used by bacteria to defend themselves against viruses. These enzymes cut DNA in precise places. We can define what these precise places are, we can know what they are, and then we can use those enzymes to cut different molecules of DNA and have them come back together based on complementary base pairing, using the same rules that Watson and Crick had introduced us to so long ago. Well, now, the second major advance that led to the current biotechnology revolution, after we had figured out how to genetically engineer recombinant DNA, was to develop the ability to introduce the DNA from one organism into another organism, creating what we call a transgenic organism. Now, this might sound like it would be a difficult thing to do, creating a kind of genetic Frankenstein's monster. And in fact, most of the technological development in this area is devoted to figuring out different ways to do exactly this, put DNA from one organism into another. But we've already discussed one common way in which transgenic organisms are created when we talked about conjugation in bacteria. Remember that conjugation occurs when one bacterial cell forms a physical connection to another bacterial cell and then transfers a small piece of DNA called a plasmid. If we use the recombinant DNA technologies we just discussed to modify the DNA in a bacterial plasmid, say by inserting a gene from another organism into that plasmid, then we can use conjugation itself to transfer that modified plasmid into other bacterial cells. Let's quickly walk through an example for how this might work. Let's say we wanted to insert a gene from a firefly into a bacterial cell. Let's put a name on this. Let's say we want to insert the gene for the enzyme luciferase, which is the enzyme responsible for making the firefly's tail light up into an E. coli cell. Why would we want to do this? Well, a couple of reasons we would want to do this. We might want to get a gene into the bacteria so that we can make a lot of copies of that gene, which we might need to do if we wanted to study something about the structure of that gene. Alternatively, we might want to do that because we wanted to make a lot of copies of the protein produced by that gene. And it's a lot easier to make a lot of the protein using bacteria as opposed to raising fireflies. Now, to do this, what we have to do is to get the firefly luciferase gene into E. coli using a restriction enzyme that will clip out this luciferase gene from the firefly's genome and also make a cut, a single cut, in a bacterial plasmid piece of DNA. If we can find the right restriction enzyme to do this, and it's very likely we can because these restriction enzymes recognize so many different kinds of sites, different kinds of restriction enzymes, then we can use that same restriction enzyme to cut bacterial plasmids, to cut the firefly genome, and then to recombine the gene from the firefly with the plasmid using the sticky ends of both to have them come back together. Now, once again, to get this process to work, there are quite a few details that we'd have to pay attention to. One of the things that we'd have to do, for example, is to make sure that we're actually putting in the right piece of DNA from the firefly. You can imagine that these restriction enzymes, because they recognize such small sequences, would cut up a piece of DNA into many, many, many different fragments, only one of which would have the gene we're interested in. So we have to develop ways of identifying which pieces of DNA we want to be part of our recombinant plasmid. We also have to figure out ways to identify the particular bacteria following conjugation that have taken up the recombinant plasmid. And this, again, involves a number of techniques that we really don't need to go into right now. The essential technique here, by the way, is usually to incorporate another gene into the same process, a gene that we can easily identify the activity of, a so-called genetic marker or reporter gene. In the case of the firefly luciferase gene, which is why I use this example, the gene itself is something we can readily identify because once we've completed conjugation, we can identify the bacteria that have taken up that gene by giving them the substrate that this luciferase enzyme works on normally, and if they have the gene, they'll light up. In other cases where we can't so easily identify the function of the gene we're interested in, we would add another gene to the plasmid. Say a gene that conferred resistance to some antibiotic, and thus know which bacteria had taken up the right recombinant plasmid and which hadn't by seeing how they varied in their resistance to the antibiotic. Now, there's another major problem that we have to overcome to use this kind of technology to express genes in other organisms that is worth mentioning in just a bit of detail, specifically when we're talking about introducing genes from eukaryotic organisms into prokaryotic organisms, like putting a gene from a firefly into a bacterial cell. As we've learned, eukaryotic genes have different kinds of mechanisms which control their expression. Eukaryotic promoters and the transcription factors that control those promoters are different from what we see in prokaryotic cells. So if we just introduced a eukaryotic gene into a prokaryotic organism, chances are good that that organism would never express the gene because it doesn't have the apparatus that would allow it to do so. For this reason, what we have to do is, if we're going from a eukaryote to a prokaryote, is not only put in the gene of interest, but also genetically engineer in association with that a promoter that the bacteria itself can work with. In other words, we have to put that gene next to a promoter that the RNA polymerase of the bacterial cell will recognize, and thus which it can use as a starting place to transcribe the gene we're interested in. There are a bunch of related problems that have to do with transferring genes from eukaryotes into prokaryotes, such as, for example, eukaryotic genes having all of these introns that normally are removed. To get around that, what we actually have to do is to create a copy of the eukaryotic chromosome ourselves that lacks these introns. We do that by taking a complete piece of RNA produced by the eukaryote and then using that enzyme reverse transcriptase. Remember that one? That's the one that violates the central dogma by creating DNA from an RNA template. And using a finished RNA template, we can then create what's called a complementary DNA strand, or cDNA, which is the gene's exons with all the introns removed. And then we put that complementary DNA into our genetically engineered organisms. Now, putting genes into bacteria is relatively straightforward because we can use conjugation. There are now many other methods that have been developed and are currently being developed to insert foreign DNA into both prokaryotic and eukaryotic cells. In general, all of these methods have in common the development of some kind of genetic vehicle, or what we call a vector, that will transport DNA into the cell in such a way that the host cell will at least express the imported genes, if not actually incorporate them into their own genome. Some of these methods involved other kinds of natural vectors, such as viruses or plasmids that are associated with plant cells. Other methods actually now involve genetically engineering small, entirely artificial chromosomes. Chromosomes that not only have the regions of DNA that we're interested in getting into some host cell, but also have all of the other regulatory DNA and other factors that are associated with normal chromosomes, so that these chromosomes can themselves be replicated and divide and function like normal chromosomes in the host cell. For example, there's been great success in creating an artificial yeast chromosome. Yeast are very simple eukaryotes, and we can then put these yeast artificial chromosomes with whatever genes we want to express into yeast cells very conveniently. In fact, there's actually been considerable progress in making a functional artificial human chromosome. Now, the technical challenges associated with creating a transgenic organism do become increasingly difficult the more complex the host organism we want to manipulate. But it really isn't going to be very long before it becomes relatively routine to insert foreign DNA into any kind of cell and to have that DNA become a functional part of the host organism's genome. There's another major advance in DNA technology fueling the genetics revolution that we don't have time to discuss in detail, but I think it's one important to mention in passing at least. This is the development of methods to rapidly determine the sequence of bases in any molecule of DNA, or as we say more simply, methods to sequence the DNA. Nowadays, when we talk about molecular genetics, we almost take for granted that we can know the exact sequence of nucleotides that make up some particular gene or other region of a DNA molecule we're interested in, like the restriction sites that a restriction enzyme might bind to, or the structure of a promoter for a particular gene. And as I'm sure you know, we've now sequenced the entire genomes of a number of different organisms, including humans. If we exclude bacteria from the list, there's probably about a dozen genomes that have been completed so far, a dozen different species. If we include bacteria, then that number goes up to hundreds. This is really a remarkable achievement and one we shouldn't take for granted. Now, as I said, we won't go into the specific details of how we sequence DNA, other than to tell you that here again, what is taken advantage of is knowledge of the fundamental mechanisms by which DNA is normally replicated in natural cells. Understanding these mechanisms have allowed the development of methods that will actually literally determine the exact order in which nucleotides are being added to a strand of DNA that's being synthesized from some template. And in so doing, with the right technology, we're essentially able to read the sequences of bases that define any stretch of DNA. Well, now that we've taken a look at some of the key techniques and innovations used in genetic engineering, I want to conclude the lecture by considering some of the ways that these techniques have been applied in biotechnology. Of course, one could teach an entire course on current applications of biotechnology, given the number and range of applications that we have today, which are increasing rapidly. What I'd like to do now, though, is just mention a few examples that illustrate the potential of biotechnology. Let me first point out, though, that actually in its broadest definition, biotechnology simply means the manipulation of organisms to make useful products. And it's something that humans have been doing since the beginning of recorded history. Selective breeding of domestic plants and animals is a kind of biotechnology. It's just that it's a very slow kind of biotechnology. What's different about modern biotechnology is that we can modify organisms much more rapidly and more radically. Now, the first commercial use of biotechnology, one that's been used actually for many years now and probably remains the most widely used application, is a relatively simple one. This is the use of this use is to manufacture particular kinds of proteins in abundance that would otherwise be tedious or costly to produce. Consider the protein insulin. This is a hormone that I'm sure many of you know is involved in the regulation of blood sugar, and people who suffer from diabetes are unable to adequately produce enough insulin. Diabetes can be treated, however, by injections of insulin. The question is, where does the insulin come from? Well, a while ago it used to be that the only source of insulin would be from farm animals, such as cows and pigs. The organs of these animals would be harvested and purified, and that would provide insulin. But that was a tedious and costly process. And furthermore, the insulin of these animals, although very similar to human insulin, wasn't identical. So it didn't always work in certain individuals. With the advent of modern biotechnology, however, it becomes a relatively simple matter to insert the human insulin gene into the genome of an E. coli bacteria, just as we talked about inserting the firefly luciferase gene. And in fact, now almost all insulin used in medical treatment is manufactured by genetically modified bacteria at a much lower cost and at a higher level of purity. There are dozens and dozens of other medically important proteins manufactured in the same way, and hundreds probably are in commercial development. Now, in the example we just discussed, the bacteria were genetically modified essentially to turn them into tiny chemical manufacturing plants, tiny protein manufacturing plants, and we were more interested in the protein, the insulin, than we were in the organism, the bacteria. But we might also genetically engineer organisms because we're interested in the organisms themselves. And many examples of this come from crop plants that have been modified. In the US, for example, close to 36 transgenic crops are now in common commercial use. Plants have been at the forefront of biotechnology because, for technical reasons, it's a little more easy to modify their DNA to genetically engineer, to insert genetically engineered DNA into their cells than it is with animals, although we can do this now routinely with animals as well. So what kinds of genes might we want to insert into a crop species? Well, we might want to put in genes that improve resistance to insects, so we don't have to use as many pesticides, and that seems like a good thing. We might insert genes that cause increased growth, or that improve the nutritional value of the crop plant. In all cases, the point is to modify the organism so it's better for our use. Well, as you're probably aware, there are many people who are strongly opposed to genetically modifying crop organisms and other food organisms. Well, why are they? Well, opponents worry about a number of issues, and these are issues worth just paying attention to for a minute. For example, if we modify a plant to include a pesticide, how do we know that that pesticide produced by the plant won't get into the environment? Or how do we know that genes introduced into modified species won't possibly get away from that modified species by some crossbreeding with a wild form? Or how do we know that the modified species itself won't escape from cultivation and essentially become some kind of super competitor with wild forms that are related to it? All of these concerns are valid, but at the same time, genetic engineering and biotechnology creating genetically modified organisms proceeds apace. It's going to be quite a lengthy debate to decide the extent to which we're willing to change species for our own use, the ethics of doing so, and the consideration of the potential costs of doing so to weigh against the benefits. Well, that's a brief tour of biotechnology and its impact. I wish I had more time to talk about it, but it's time to move on. And in our next lecture, we're going to come back to our theme of development and homeostasis and begin to look at the process of how cells talk to each other.